Okay. So good evening to all of you. Good evening, Juveria. So my voice is audible to you, Juveria? My voice is audible. Okay. So yesterday we have done the chromatography techniques. So are you able to understand it basically? And exchange chromatography, or we also discussed some about paper chromatography or column chromatography. So are you able to understand it? Good evening, good evening. If my voice is audible to all of you. Is my voice is audible, students? So today we have to do flow cytometry. Yes, Komal is here. Good evening, Komal. So are you getting my voice? Just waiting for some more students to join, then we will start the session. So if you have any type of query or if you have any type of question, then please let me know. We will discuss it, or we will discuss when other students will come, then we will discuss. Kindly let me know if you have any query, or are you able to get what we have done yesterday of chromatography techniques basically. So Devashish is here. Good evening, Devashish. Michelle is here. Good evening to you. So is your college open today? Have you classes today also? Or are you free in the morning? So the area, Komal, are you free in the morning, or you have a classes in the morning also? Because this is Saturday today. That's why I am asking. Kindly reply. So do you have any type of classes today, or are you free in the morning? Are you free in the morning? Yes. I'm saying, are you free in the morning, or do you have any type of classes in the morning, like the college session, the college classes, or are you free in the morning? Is I am audible, students? Are you getting my voice? Are you getting my point? Kindly reply, please. Should I send the this link again in the group, or is it okay? The students will get it. Yes, we have lecture in the morning. Okay, 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 okay. But tomorrow is Sunday. Are you free? Are you free tomorrow? Because I have to take your classes in the noon. Tomorrow I will take it in the noon, and maybe for more than 2 hours. So that is why I am asking that tomorrow because tomorrow is Sunday basically, so you have no classes from the college. So if you are free, then we will take the classes in the noon. Okay, Devita is here. Yes, ma'am. Okay, 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 okay. You have classes today. Okay. So on Sunday, that is on tomorrow, we will take your classes in the noon. So that's why, so that you will become free also in the evening session. Like from 4 to 7 we are here, and we can't perform any type of activities in the evening. So I think we will take the class in the noon after lunch basically, like from 2 to 5 or 2 to 6, or I don't know how long it will take, but I think we will take it in the noon after the lunch. You suggest the time in which timing you are comfortable. I thought it should be after the lunch only, like from 2 p.m. Are you comfortable from 2 p.m.? So that we will wind up everything before 5 p.m. That's my thinking, that from 2 p.m. we will cover everything in between 4:30 to 5 p.m. So by this timing, now at this timing where we are joining, you will be free at that time. So is it comfortable to all of you? Kindly let me know. From 2 p.m. yes, after lunch basically. So after lunch, that is around 2 p.m. we will join, and we will wind up everything in between 4:30 to 5 p.m. Kindly let me know. Komal, Devashish. Where are the other students? They have not yet. They are not here yet. GT is here. GT is here. Okay, good, good. So we will wind up within 4:30 to 5 p.m. Gayatri is also here. I'm just waiting for more students to join, because I want to tell you something, I want to communicate with you. So just waiting for 2-3 minutes more so that all will join, and then we will start our session. Saurabh Shinde is here. Saurabh, I found your message only today. Are you there in the before classes also? Saurabh Shinde. Did you attend the previous classes also? Saurabh? Yes, today we have to do a very important topic, which is flow cytometry, the very recent technique basically, and I will try my best to convey it to you. So Saurabh is saying, yes ma'am, I was there. Okay, good, because I found your name or comment only today, that's why I am little bit confused. Prachi is also here. Prachi is saying good evening ma'am, good evening to you. So flow cytometry is a very recent technique and little bit complex also, but not as that much. We will definitely do it best. So where are the other students? What they are doing? Students, you must know that you have to write a review paper. Are you aware of it? You have to write a review paper. Siddhi is here. Good evening, Siddhi. Amita is also here. Amita, good evening. So, yes, Cloris is also here. Good evening, Cloris. Where is Rutuja? She is here yesterday. I am not finding it her now. Anushri is also here. Where is Rutuja? Rutuja, if you are here, kindly reply. Komal is saying good evening to all. Okay. So it is for all of you. Rahul is here. He is saying good evening Rahul. So I am just asking, are you aware of it that you have to write a review paper which will be published in a journal, and it will become your activity, or it will become in your daily routine that you have published one paper in a journal. So did you aware of it or not? Aditi is here. Good evening Aditi. Gautam, good evening. Where is Rutuja? If you are here, Rituja, kindly reply. Okay, so all of you, do you know that you have to write a review paper also? Did you aware of it? No. Okay. Because we have now discussed it in our previous classes. I think I have to tell it to you in the second class, but I think you have know about it, so that's why I didn't convey the message to you. But it's okay, no issues, no worries. We will discuss about it today. Okay, no issues. We will discuss about it today, because it is a part of your this lecture series, or this MBBT series, in which you have to write a review paper. So Vatsal sir asked me to provide you any topic, but my point of view is that you are young generation, you are quite mature. You can select your own topic. You can select any topic which is your field of interest, any topic. Like you can write about this pandemic which is happening these days, like COVID-19. You can write, and you have to make it a review paper. So basically we have types of paper. We have review paper and we have research paper. In the research paper is generally for those students who are pursuing their JRF, SRF, or you can say PhD. So during their research work, what they do, they put their research in the form of a paper. They put their research, like if I suppose I have done work on mitochondrial DNA. So I will put my research in the paper, so that all the another person throughout the world, all the scientists you can say, or the researchers which are present throughout the world, they are able to get my research. They are able to keep in touch with me. So this is a research paper. But as you are very young now, you are not doing any type of research, or you are not in JRF SRF, so you have to write a review paper. Now what's a review paper? Review paper is basically what in which we review the paper of another person. Suppose I have taken the topic, any topic, suppose I have taken cancer, I have taken breast cancer, so I have to write in my paper only the work which is done by the previous researchers. The last 20 years, within the last 20 years, the work which is done on breast cancer, so we have to write it in our review paper. All the information will come in a single review paper. So the person, when I am here, now I am reading the review paper of Anushri. So Anushri has written one review paper in which she has added the research of last 20 years, research of people from last 20 years. So I have not to go to all the person's paper. Instead I will read Anushri paper and I will get all the information. Are you getting my point? Buffering. Oh. 1 minute please. It's okay. One minute please, 1 minute. Are you getting me now? Are you getting me? Shall I speak? Okay. So I don't know where I was before, so I will start once again. So in a review paper, suppose I am reading the review paper of Juveri, or I am reading the review paper of Saurabh. So Saurabh has included all the research which has taken place in the last 20 years. So I have not to go individually to all the scientists or the researchers. Rather, I will read only Saurabh's review paper. So this is your duty, or this is the work you have to do. You have to search any topic. It's up to you, because you can do research or you can review paper on any topic. Like these days, as we have this COVID-19 pandemic, so you can also write the paper on COVID-19, because this is very recent. You also know about corona, the virus, and from where it starts and how it comes to India, then how it comes to our own region, our own locality. So I think this is very easy for you, as it is your first attempt. I don't want to make it little bit complicated. Or you can write it on breast cancer or cancer technologies, or you can tell us that how the technologies related to cancer now get more and more better within these 20 years. You can write it on breast cancer. You can write it on CRISPR technology, for which the scientist got the Nobel Prize. So it is up to you. So yes, it's compulsory, I think. So that's why Vatsal sir has told me yesterday only to ask the students to write a review paper. Yes, to get the certificate, and even this will become your achievement also, that you have a research paper, you have a review paper. It will enhance your CV or your resume. Yes, it's compulsory, because that's why Vatsal sir has told me yesterday only. One student of you actually asked me, then I asked sir, and he said yes, we have to have to collect the review paper. So you have to write a review paper and then you have to mail it to me. I am there for helping you all. No issues, no worries. The only thing which you have to do, you have to write it on a MS Word. Then you have to send it to me. I will make all the corrections. I will make it, you know, I will make it okay for your journal publication, and we will select the journals which are basically free. So you have to send your paper to those journals. Okay. So I don't know how much of you know about a review paper or a research paper. Tomorrow we will again meet, then I will show you some review papers or research papers that we have a title of it, then we have to write a small abstract, then we have to write introduction or material methods or review of literature. So today is only you have to select your topic only. That ma'am, I want to write on this topic, I want to write on ovarian cancer, I want to write, I don't know from which stream or which is your point of interest. That is why I am not bonding to a small topic. It's only your interest. You can write it on anything. You can write it even on molecular markers. You can write it on these biological techniques. You can write it on Western blotting. So you can write it on any topic. Actually, okay, I will talk with Vatsal sir again. He only mentioned me to ask you to write a review paper, and it's not a big deal. You can write it. You all are a brilliant students. You can write it. So today I just want you to select any topic which is your point of interest. You have to select it only. Then I will guide you further that how to write it and in which journal we have to send it. Okay. So I don't know really about that thing, but I only know that you have to write a review paper either for the Khalsa college students or for the internship students. I don't know regarding it. I will talk with the sir first, then I will let you know. Okay, so Gayatri is saying that it is only for internship students. Okay, this is okay. Actually, I am not aware of it right now, so I can't say anything. Okay, if it's for internship students, so for those students you have to write a review paper. You can write it on any topic. Yes, Devashish is also saying yes, Gayatri is saying right. So it is only for internship students. It is only for internship students. Okay, I will share my email ID. So those students who are from internship program, so they have to send it to me. I am sharing my email ID, so you can send your review paper to me. And about the time limit, actually this web series is for 21 or 22 of October. So within this time period you have to submit it. Are you comfortable within this time period? Hanskrita, are you comfortable in this time period? Or you need more time? So this is my email ID. You have to send it in. You have to send it here. So where is Hanskrita? Okay, suppose I want to conclude this talk with one little more advice. That suppose if you are writing a review paper on COVID-19 pandemic, if you are writing it for this, more time, ma'am. Okay, you need more time. Try to conclude it within 25th of October. 25th of October. Or okay, it's okay. You have to conclude your review paper and send up to me by 27th of October. So today is 17 and you have 10 days with you. That is 27th of October is the final date of submission. So are you comfortable now? Are you comfortable now? So to conclude this whole talk with one more important point. Let's suppose if you are working on COVID-19 situation or you are discussing about pandemic situation, instead of writing only a review paper that what is this or what is this, you can add the data in it also, so that lots of people are there, lots of people. Okay, good. So lots of people are here who are interested in knowing all about this pandemic. So we will collect that data. So you have to start it that it starts in China from this date, then it becomes pandemic in China in this date, and how many people are died in China at that time. Suppose if it is in December 2019 in China, so you have to tell that on this date it becomes the China declared pandemic in this date, and that many people died at that time. Now you have to tell in this date it introduced into the India. Within India you have to tell, like we have all the data from January to up to now, that in March we have so much of infection, in April we have so much of infection. Even you can also mention, if you are only specifically writing about India only, so you have also an option that you can write that in India, so this is our lockup, you can say. So we have this first round, second round, third round of these all things, where we have not to allow to do any type of social gathering. So you have to tell it even in 2020, you can mention it in the months also, like in January we have so much of infection, we have this thing, and in March, April, so it will become your data, and your paper will become so important that so many people will read it. Basically thing is only that if you are writing any type of paper, how many people are reading your paper, this is only important. Then your data will come under Google Scholar, and if the people will type your name. Suppose you have Sejal here or Hanskrita is here, they will type on Google Hanskrita, and what will they find? They will find their review paper or a research paper, and all the persons or all the people will read their paper. So your first paper, even you will take more time, it's okay to take more time, but your paper should be more authentic. So instead of only copy pasting it from the others person's work, rather you perform your own work. You just collect your own data. Like if you are in Mumbai, so starting from India, that in India this thing happened within this month, then after it you can also come up to your own town. So you can also write something about your own city, like in Mumbai, in 1st of March or 1st of April, that much type of infection is there, and now, so lots of things are there for you to write it up. Even I recommend all of you, whether you are in internship, whether you are in this series, I don't know regarding it. So I just want to request you all that please write something about this pandemic. You will get lots of people who will read your paper. Even if you can type your name on Google, your paper will come, and it will become highly effective for your future perspectives. If you want to go in research, if you want to go further for PhD, for JRF, for SRF, for that reason you have to write a paper, and I am here with you for helping you all. So I just request all of you that please write something about this corona pandemic or about this, so that we will collect all the data which we have, even from January, even from February. So are you getting my point, what I want to convey? Are you getting it? Are you getting it, students? So it's my request only to all of you that please write something. You have, because now we are in home, we have lots of time. So we have lots of time, we can write it. Are you getting it, students? Are you getting it, बच्चे? समझ आ रही है? Okay, good. So I request all of you to write something where you have some type of data. You can even if you don't want to mention it about any other thing or any country, you can mention it only about your own city. So the person or the researchers who are there in your city, they will definitely read your review paper. They will definitely read your paper. That's my guarantee. Even they will use your paper for their own research or for their own data. So you have to write something. Even if you are not want to write about your own city or about Mumbai or about India, then you will collect the data from all other countries. Like you can mention it, like in Switzerland how much pandemic is there, in USA how much pandemic is there. So you can compare it between different countries also. It's not only within India, it's not only within our own cities. No, you can do the comparison within the countries also, within the facilities which are provided within the country, the facilities which are provided in Canada or which is provided in USA. So this type of paper is also, if you have this type of comparison within different countries, then this review paper or this article will be read by foreign persons also, or foreign researchers who are pursuing their PhD. So it is up to you how to make it. You can compare it within India also. Within India you can compare between different states within India, or you can compare within different countries like New Zealand, we have Switzerland, we have different countries, China is here. So this is the latest topic which is very prominent nowadays. That's why I am focusing to write on this topic only. Otherwise you have cancer, you have breast cancer, ovarian cancer, you can write it on also. It's up to you. But I think COVID-19 pandemic is the one thing which is there in our life, so we have to know about it very briefly. So are you getting it now? I hope all of you, whether you are in internship or whether you are in series only, I hope, or you can also do one more thing. Those students who are not in internship programs, like they said no need of it, so you can collaborate also. Like Gayatri is here, Jeet is here, and other students, you become a group of 5 students, 4 or 5 students, you can collaborate because you are from the same college basically. So you can collaborate together and write one paper. Are you getting my point? It is for those students who don't want to write a paper, but I emphasize them, I just requesting all of them to make a group of 4 or 5 students. You are from the same college, so even you must be in contact with each other. So you make a group of 4 or 5 friends or 4 or 5 students, and just assign the duty in between yourself, like you have to collect the data from this city or this country, and then just do the collaboration. Is it a good idea or not? So it will increase your knowledge, and it is for you only. I am there for helping you. You have to just collect the data. The data is there in our newspapers, in the Google. The data is everywhere. We have to just compile it only. Yes, it is going to be published. Yes, and my priority is basically to put it in free journals, the journals which will not pay anything. So that is why I am asking, just prepare your mind, prepare some topics so that we will pursue it further, and you will get your name on your paper, and it becomes very big achievement when you are in bachelor's only. So I think it will also help in your master's degree or in any type of selection. It will help you definitely. So Sarkar Raj is saying something, but I am not able to understand from these emojis. So kindly let me know what are you asking basically. That's confusing. I am not understanding the emojis. What are you saying? Please write it down, Sarkar Raj. I am confusing. I am not getting it. Sorry. So are you able to get my point? So Gayatri, is it okay, bache? Gayatri, Prachi, Jeet, is it okay to all of you? Are you comfortable now? Kindly let me know, please. Debashish, Komal, Lai, Amita, Siddhi, Riya, kindly reply, bache. And Sarkar Raj, I am really not getting your emojis. Kindly let me know what you want to say about these things. I am not understanding it. Okay, kindly let me know. Okay, Jeet, say yes, ma'am. Okay, good, bache, very good, very good. So just try it. Every time we fall it little bit. Every time we feel it little bit difficult when we have to do new activity, but it will be very good for you. I really it's my guarantee that it will help you in future. I promise you regarding it. So okay, they are getting my point. Okay, very good, very good. Tanmay is also here. Tanmay, how are you? Sakshi is here. Okay, Debash is saying currently all the students are very busy. Okay, Debash, no issues, no worries. Just try your best. If it is possible, then most welcome. If it is not, then it's okay. No issues, no worries. Just try it once. If you can, then I think you can. You will do it. So just try once. Only from my point of view, just do only once. If you are comfortable to write it. I know you all are busy because of your busy schedule. Even in the college classes are there. Then we have this 2 hour class. So I know. But try only once. So all are you getting my point? Okay, very good, very good. So if Rituja is friend of someone of you, after this class you have to tell her that ma'am is asking about you, because I am not getting her right now. So you have to convey my message to Rituja. Whose friend is Rituja basically? Sakshi, is it your friend? Yes. Devita, okay, bache. Just try, only just try. Jeet is the CR, okay. Jeet, what to say to Jeet basically? Ask the CR of Khalsa. So Jeet, kindly put my message to Rituja to where she is today and ask her to join the session. Okay, that's the only thing. Just Divya is also saying we have our practical assignments. Yes, she has lectures and practicals and assignments and all. Okay, no issues. Just try once only. Make a group of 4 to 5 students from your own class. If Jeet is here, Jeet, make a group of 4-5 students and send me a list that these are the 5 students. So if you write even one paragraph, if you write one or two paragraph, you are able to compile it. So Jeet, where is you? Kindly let me know. You actually, Jeet, if you are a seer of a class, after this class, kindly interact with your friends, with your students of the college, and let me know. Are you getting my point? What I want to say? After this session, just talk with them. So you don't need to worry about anything. There are no issues. Even you can make the review paper of 3 or 4 pages only. So you not have to make it for 10 or 15 pages. No, it is only your first assignment or your first review paper. So 3 or 4 pages are okay also. I am not forcing you to write for 14 to 15 to 20 pages. No, if you can write 5-6 pages, these are more than enough. Ma'am, Jeet is the CR of SY. Okay. And we don't know about TY. Okay. So if the CR is here, he will discuss all the things with you. If you are comfortable to write 3 or 4 pages or 4-5 pages, then it is very good. If it's not, then it's okay, no issues. Okay. So that's all for the review paper or the article which you have to write. Now we will conclude this thing up to here. Oh, Rituja is here. Rituja, you are not commenting anything. You are not writing any type of message. That is why I am thinking you are not present today. Okay, how are you? Are you comfortable? Are you okay today? I just only remember your name from yesterday conversation. That's why I am finding where is Rituja. Otherwise everything is fine. Okay, no, don't worry about anything. So with this we conclude today's this article session. We have to wrap it up because we have to go further to the flow cytometry. So is it okay? Shall we move further? Yes, now there are many students are now joined today. Juveria is the CR of TY. Okay. So Juveria, you are, if you are the CR, then kindly convey my message to other students which may be not here today, or if they are here. So you have to communicate with them. You have to talk with them, and you have to make the group of 4-5 students, and then you have to write the review paper of 4 to 5 pages. If it is possible, then it's good. If it's not possible, then it's good also. But please kindly let me know only. Okay. So are you getting my point, Juveria? Are you getting my point, bache? I think she will get. So with this we are concluding this review article or research article talk. Now we will move further to our yesterday's topic, which is basically chromatography. We have done paper chromatography, column chromatography, ion exchange chromatography. Then we have gel filtration chromatography, and we have only one thing which left is the affinity chromatography, which we will discuss today. Before it, yes, 3 people group is also okay. Yes, even you can write it individual only, everything is okay. But those students who are little bit busy these days because of these assignments or lectures, so they can make the group of 4 people or 5 people, but not more than 5 people. 3, 4, 5 is comfortable. 3, 4, 5, 2, 1, 2, 3, 4, 5 is okay. Just try to write. Okay. I hope you will get my point. So I have one more announcement which we have to do today. That tomorrow we will take the session in the noon from 2 to 5 p.m. basically. That means within this time period we will become free. Yes, very good. So Juveria get my point and she will convey the message. So you have to also, Jeet and Juveria, you also have to convey the message to the students that tomorrow we have a class on 2 p.m. That is after lunch we will take the class, and by this timing, by this 5 p.m. we will be free. So from 2 p.m. to 5 p.m. we will take the class. Maybe we will take the break in between of 5 minutes or 10 minutes. I don't know regarding it anything right now. We will see it here. We will see it tomorrow. So these are 2 things which I have to discuss. If you have any type of question, kindly let me know. Or now we will start. It's now already 5:30, and we have to do flow cytometry today also. Ma'am, I am class at that particular time. Oh, Manpreet, on Sunday also you have a class. How long your class will take and what is the timing of your class? What's the timing of your class, बच्चे? What's the timing of your class? Is it from 1 to 2, 2 to 3? It's from 3 p.m. From 3 to 4 p.m. Your class is from 3 to 4 p.m. Manpreet. Three to 5 p.m. To our class, then how will we will manage our class, because I think we have to do it in the noon so that we will be free by this 5 p.m. So is any other student has the class on the same timing? Please let me know if any other student has the class in the noon, tomorrow noon, that is from 2 p.m. to 5 p.m. or 1 p.m. to 4 p.m. like that. Rituja, Jveria, do you have classes on that time? I have to leave one particular class to attend this class. Actually, I don't want that you leave any class. I want that you will join both the classes. But let me confirm from the other class also whether they are free on this timing or not. So Jveria, kindly let me know, and Jeet also, Devashish, kindly let me know whether you have a class in the noon, in tomorrow noon or not. No ma'am. Priya is saying no ma'am. Okay. Rahul, kindly reply. Saurabh, Devita, and where is this one, Sarkar Raj? Where is you? You are not even telling me the meaning of these emojis. Where are you, बच्चे? Kindly reply. Are you free on tomorrow noon? Sarkar Raj. We have Gautam here. Riya is here. Sakshi is here. Rahul, Siddhi, reply बच्चे. No. We are free only on Sunday. Okay. Good. Saurabh saying no ma'am. Okay. So Manpreet, we will. Okay. Sarkar is saying that he is free maybe on Sunday. He is saying thumbs up. Okay. So Manpreet, we will try our best to reschedule the event, but it may be from 2 to 5 p.m. बच्चे. So either you can see the recorded session on YouTube, or I will discuss with you individually also. No issues regarding it. If you have a class tomorrow, take that class. If it is important class, take that class. I will discuss everything with you individually also, some other platform, or you can watch it on YouTube, recording also. So I hope you will understand the whole thing, that tomorrow is the holiday and I want to wrap it in the noon. So if that class is important, the another class is important, you will take the another class. No issues at all. This section which we will do today, I will make it available to you again. So we will discuss it again either on Google Meet or some other platform. So no issues regarding it. So Manpreet, are you getting my point? Kindly reply me. Manpreet, if you are getting my point, if that class is important to you, kindly take that class. This class we will discuss again. No issues regarding it. So are you getting my point? Kindly let me know. If any other students, 2 or 3 or 4 students who are unable to take the tomorrow session or tomorrow class, then we will discuss them on some other platform like on Google Meet or on YouTube, something like that. We will discuss it again. Yes, okay, very good, very good, very good. So if you have some issues for tomorrow, then we will discuss it again. So with all these things, now I am concluding the conversation round. Now I have to complete flow cytometry, and before it we have affinity chromatography which we have to discuss. So now I am starting the chromatography. So this is the chromatography which we have done yesterday. I will just revise it very fast because we have done it before also. So what are proteins? These are the biomolecules which are made up of amino acids, and they are the building block of our system, and the proteins perform most of the biological functions of the system. So proteins are the ones which are made up of amino acids. They are the building block of our body, and they perform the biological functions of the system. So whole biological functions are performed by these. Most of the biological functions, not whole, but most of the biological functions are performed by these proteins. So this is that how they are made. They are made from the polypeptide chain by amino acids. They are made. These are the types of protein which we have discussed: fibrous protein, globular protein, and membrane protein. So the example of fibrous protein is collagen, which is insoluble in water. You have to remember this thing, that they are linear structures and they are insoluble in water. Then we have globular protein, which is actually little bit complex as compared to the previous one, but it is soluble in water. If you can see here, so it is soluble in water. So it is water soluble. Now what happened in this case? The hydrophobic interactions are inside. The hydrophobic chains are inside, and the water loving hydrophilic side chains, they are present on the outside. So that is why they are water loving or water soluble. Then we have membrane proteins, which the example is bacterial rhodopsin. These are also hydrophobic in nature, and they are water insoluble because the hydrophobic interactions are present outside and the water loving interactions are present inside. So these are the types of proteins which we have discussed. Then we have the structure, like we have primary structure, which is very simple. It is just the amino acids which are formed by peptide bonds. Then we have secondary structure, which is complex. We have tertiary structure, which is more complex as compared to the secondary one. And the last is the quaternary structure, in which we have 2 or more polypeptides. So 2 or more polypeptides are linked together through covalent and non-covalent interactions. You have to remember this thing also. Like in secondary structure, we have every type of interaction. We have peptide disulfide interaction, non-covalent interactions like H bonding, electrostatic, and non-covalent interactions. And in quaternary again we have covalent and the non-covalent interactions between 2 different polypeptides, between 2 different, not only 2 or more polypeptide chains. So this is a protein. This is a basic primary protein. You can see the amino acid leucine, alanine, glutamine, valine, leucine, tryptophan, leucine, valine. So they are joined together by the peptide bonds or disulfide bonds. Then we have a secondary structure, then a tertiary structure is little bit complex. Then what is protein? We have done yesterday protein, how to do protein isolation. We have different types of methods. We have different types of buffers like lysing buffers, homogenizing buffers. So this is the extraction or the isolation of the protein from the leaves. Okay, so then we have to perform it in an ice cube. I also told you yesterday why we have to put it on an ice cube, because we don't want to damage their primary structure. We don't want to damage because of this excess treatment of adding the salts, adding the buffers, and then we have to crush it. So we don't want that the basic structure of proteins get damaged. So we have to do it under very low temperature. So this is again centrifugation is there. Then this is, if you can see, the lower one is the sample, the upper one is the supernatant, which we have to throw out, and the sample we have to take it for further actions. So this is these things we have done yesterday. So this is the extraction from fungi. So we can collect protein from everywhere, from plants, from animals, from fungi, from any living substance. So this is the procedure of that thing. I will not discuss it now. The SDS page we already done it. The only thing which we will do, which we will revise, is the staining and de-staining solution. So in SDS page, which is the staining dye, it is Comexi Brilliant Blue, which is a dye which is used as a staining solution. Then we have a de-staining solution. For what purpose we have a de-staining solution? We have a de-staining solution to remove the excess stain, so that we will be able to see the bands clearly. So these are the bands which are there. This is the SDS apparatus. This is the SDS page by using silver staining, or not Comexi blue but another type of dye. Then we have spectrophotometer, which is basically used to know the concentration of the protein, how much concentrated or how much good amount of protein we get. So you have to remember the absorption ratio of protein is from 280 to 200 nanometer. That means between 200 to 280 we have the absorption of the proteins. So this is the procedure of your spectrophotometer. Then we have to do protein purification. So the one thing which came in my mind, why we need proteins, why we need to extract protein, why we have to purify the proteins, because we want to study the structure and function of proteins. I isolate that particular protein. Why I isolate that particular protein? Because I want to study the structure and function of that particular protein in the body of any individual, or from where we have taken it. Or we have to study protein regulation and protein interactions. So these type of things, for these things we have to need protein which is properly isolated and properly purified. So isolation, separation and purification is the most important step for all these activities. For any type of protein regulation, for any type of studying interactions or producing antibodies, we need proper isolation, separation and purification of the protein molecule. So these are things from where we can get the protein. We can take it from the tissue, from liver, from anywhere, or we can also take it from the cells of the body. So these are the methods of protein separation or purification. We have homogenizer, we have centrifugation, in which we separate the pellets according to their molecular weights. Sorry, I am doing it little bit fast because we have done it yesterday also. So these are the techniques for the isolation of protein. We have salting out, we have dialysis, which we have done yesterday. We have gel filtration chromatography, ion exchange chromatography, gel electrophoresis, affinity chromatography and isoelectric focusing. Isoelectric focusing is a part of 2D gel electrophoresis basically. The thing which I also told you before, that if the molecular weight of protein is same, then how we will separate it on a SDS page. So it's a part of 2D gel electrophoresis or isoelectric focusing. I will definitely discuss it, but if I forget, then you have to remind me that this one thing is left, that if the molecular weight of proteins are same, suppose we have 25 kilodalton, 25 kilobecdalton, 2 proteins. So in the SDS page they will show band on the same point. So how we will recognize that this is the different protein, this is a different protein? If they have the same molecular weight but they are not able to distinguish by SDS page, then we have another technology which is known as isoelectric focusing or 2D gel electrophoresis. It is just the higher version of the SDS page. So we have the purification techniques like dialysis, centrifugation, this size exclusion or gel filtration therapy, isoelectric point salting out, ion exchange chromatography, and electrophoresis. We have discussed all these techniques. This is dialysis. We have semi-permeable membrane. Only the small particles are allowed to pass through it. If you can see here, this is a semi-permeable membrane, and look at the size of the pores. So only the small particles, only the small protein molecules are allowed to pass through it. Then we have centrifugation, in which we have to run it under a very high speed or under a very high RPM, and the heavy particles will settle down and the lighter particles will be on the supernatant or on the upper stage. Like here you can see, this is 60% sucrose, this is heavy. So after centrifugation it is again present here. But this is 20% sucrose, it is quite heavy from this gradient. After centrifugation what will happen, it will settle here just above the 60% sucrose, and on the upper we have only supernatant. Then we have chromatography, in which we have to separate the proteins or the mixture or the compounds in order to analyze, identify, purify the mixture. So we have to do these things. We have to analyze it, identify, purify and quantify the mixture. In it we have done gel filtration, ion exchange, dialysis. This gel filtration you already know about it, that this black one are the beads basically, which are porous in nature, which is made up of gelatinous substance, and they will absorb the small particles. So small particles get embedded in. If you can see here, you can see this is porous beads, and blue one, the blue particles will come inside, and the red one, the size of red one is quite large, so they will not come inside and they will move as such from the apparatus. So this is the gel filtration, that we have the resins which are made up of gel, and the small molecules or the small proteins will come inside in this resin, as it is porous in nature, but the large one are unable to come inside. They are not able to come inside. So what will happen? It will go as such from the chromatographic apparatus. So it will not move into these gel beads, or you can say resin beads. Then the another thing which is very important is ion exchange chromatography. I asked you again and again that kindly make some type of notes or write it somewhere, because otherwise we will forget it. So you have to remind that thing. We have two types of exchanger. We have cationic exchanger, we have anionic exchanger, and it's a property of exchanger that if it is suppose here we have cationic exchanger which carries a positive charge. If it is a cationic exchanger, it will carry a functional group along with it. It will carry a functional group along with it, which may be a CM group. So it is. So we have this apparatus. We have this one. Suppose this is a cationic exchanger. So beads are basically positive in nature, and they will carry a small functional group which is negative. So we have a small functional group which is the CM group, and it is negative. So this is the first step, which is the preparation of the chamber or the preparation of the apparatus. You can see here, this is the first step of equilibration. So now you have prepared it. After it you have to add the solvent, in which we have our own desired proteins. So what will happen then? So this is the thing, we have cationic exchanger with small functional group which is basically negatively charged. Now this negatively charged protein, what it will do? It will attract the positively charged proteins. So if it will attract the positively charged proteins, the negatively charged proteins will go as such. They will go as such without any type of combining. So the cationic exchanger, in the cationic exchanger we have a small functional group which is negatively charged, and this negatively charged small functional group will attack or will attract the positively charged protein molecules and allow them to bind with it. So what will happen basically? The positively charged protein will be bind with it, and the negatively charged proteins will move as such. I will show you in the diagram. So here you can see this functional group, this is the CM functional group. This functional group allow the positively charged group to remain with me. So this functional group allow it to remain with me, and these negatively charged proteins will move through the column as such without any type of attachment. Okay. So now when it will attach, these red protein molecules, they are washed out. Now the second step is what? Now we have to take the negative protein molecules are washed out. The negatively charged protein molecules are washed out. Now what we have to do? We have to separate this positive one also. So what we will do? Now we have to increase the concentration of elution buffer, which is basically the sodium chloride. So we have to add sodium chloride. When we add sodium chloride, what will happen? This CM group or negatively charged group now attach with this sodium, because it is negatively charged, so it will attach with this sodium molecule and they will form a group, separate group, and this desired protein which we want, it just attached with the chlorine molecule which is negatively charged, and now we can separate it from the column. So this is the basic technique of ion exchange chromatography. Are you able to get it? We have a cationic exchanger which is positively charged, but this cationic exchanger takes a small functional group which is negatively charged. So this negatively charged molecule will attract the positively charged protein molecule. Now they will bind. After it, negatively charged molecules or protein molecules will be washed out. So what we have to do? When they are washed out, the another thing is the addition of the elution buffer. In the elution buffer, we have the excess quantity of sodium chloride. Sodium will attach with the negatively charged CM group, and chlorine, which is negatively charged, will attach with the positively charged protein molecule, and by this technique we are able to separate the negatively charged molecules first, and after it we are able to separate the positively charged protein molecules. So this is ion exchange chromatography. So here you can see first negatively charged molecules will be moved, and then we have the positively charged protein molecules which are extracted. So these are the functional groups which we can use, like DEA cellulose, we have CM cellulose, we have DEAE which we are using in case of anionic exchangers. So these are different types of exchangers. You have to remember only 2-3 only. So this is the ion exchanger. I hope you get it. Gel electrophoresis we have done, page we already done before. Now we have affinity chromatography. But before affinity chromatography, kindly let me know, are you able to get anionic ion exchange chromatography? Are you able to get it? Kindly reply me fast so that we will move further to the affinity chromatography. Yes, ma'am. Okay, good. Good. Very good. It is only the revision of yesterday topic which we have done, but I just want to do it again so that it will just put in your mind and you will not forget it. Who is aloo paratha? Aloo paratha? Okay. So now we will move to the another important technique of chromatography, which is affinity chromatography, and after it, these chromatography technique will be finished. We have Siddhi here, Vaishnavi here, Gayatri, Prachi, we have Juveria is here. Okay, good, very good, very good. Rituja is here. Okay, now we will move towards the affinity chromatography. It is same as we have another chromatography, but we have to cover it also. So let me conclude it with this affinity chromatography. So what is basically affinity chromatography? That proteins are passed through column of beads containing a So what do you mean by column of beads? Like this is a column. So we have beads here, which may be gelatinous in nature, which may carry any charge according to that chromatography. So proteins are passed through a column of beads containing a covalently bound high affinity group for the protein of interest. Like in ion exchange chromatography, we have positive and negative ion interaction. So in affinity chromatography, we have an interaction between, like I will tell you the interaction. Bound protein is eluted by a free high affinity group. We have interaction between the different affinity groups basically. Let me tell you. An antibody specific for a protein is immobilized on a column and used to affinity purify of specific protein. So we have a antibody protein interaction like here. So it has a specific binding properties. The polymer, which is a stationary phase, is covalently linked to a ligand that binds specifically to a desired protein. You all know about antigen-antibody interactions. We have a ligand here which binds to the specifically desired protein. The bound protein can be eluted by adding high concentration. So after when they have the bonding, the excess protein molecules will be washed out and the desired molecules are then removed with the help of elution buffer. So this protein ligand interaction can also be disrupted with a change in a pH. So this is also a elution process. This is also a elution process. But before elution, we have to do the protein and ligand interaction. We have to do the interaction. Like we have antigen antibody interactions, histamine and this interaction, GST and another tag interaction. So I will show you in this diagram. Yes, so these are the, if you can see, the beads are attached with the specific ligands. The ligands have specific shape. If you can see, the ligands have this type of V shape. So only the molecules which have this type of structure, like the molecules are square in structure, they are rounded also, they are triangular also. So only the molecules which are triangular in nature, they will bind specifically to the ligands. So they will bind specifically to the ligands. This is beads only. These are the beads which contain the ligand. So what will happen? Only the triangular one, this triangular one, they will bind with the ligand, and the another protein molecules which are square or which are rectangular or circular in nature, they will move out first. So they will move out first and we have the interaction between the protein and the ligand. So now we have only this, our desired protein is here. So here we have only our one desired protein which is triangular in shape or which is triangular. So now we have to add the elution buffer, that is we have to add the high concentrated buffer in it to break this interaction and we will able to collect our desired protein. So this is the protein separation. We have 2-3 types of protein, but the desired protein is separated by with the help of elution buffer. So this is affinity chromatography. So you can see here also in this column. So this is the beads which have a specific pattern, or you can say specific ligand structure. So we have ligands on which specific protein molecules will bind. As you can see here, only this red one which are hexagonal in structure, they will bind, and the rest of the other which are triangular, which are circular, they will washed out. Okay, so this is number step. One step is loading, number 2 step is separation. So these molecules will be washed out and we have only the desired protein molecule which binds with the ligand. Okay, so the non-bound samples which are not allowed to bind or which have not a particular shape for binding, they are washed out. And in the third step we have the elution buffer. When you add the elution buffer, what will happen? These molecules, the desired molecules are also break from the ligands and they will be collected in a separate column or in a separate tube or a vial. So this is our binding affinity or affinity chromatography, in which we have an interaction with, like we have a ligand, a protein ligand interaction. So this is the binding affinity or affinity chromatography. It is, if you understand ion exchange chromatography, then it is not difficult for you, because it's only a simple thing. So like if we have an enzyme as a ligand, so the target molecule may be substrate analog, inhibitor or a cofactor, or you can also antigen antibody interaction. So in the column you can have antibody as a ligand, and antigen will definitely come to attach with it. So this is the antigen antibody interaction. You know about this enzyme ligand interaction. So like we have hormone and receptor. So we will add a hormone as a ligand, and the receptor will come to bind with it. So these are only the type of interactions. Like we have this glutathione, and this is the GST fusion protein. So what will happen? This GST fusion protein will bind with this glutathione, and the rest other proteins will be washed out, because GST fusion protein has a specific structure with glutathione as a ligand, and they will bind together. So if we have glutathione, what will happen? GST fusion protein will bind with this glutathione. So these are the interactions. You will get it. Like if we have a lectin as a ligand molecule, what will happen? It will allow the polysaccharides or a glycoprotein or any type of receptor to bind with it. So these are the interactions. We have enzyme substrate interaction, we have the antigen antibody interaction, we have hormone receptor interaction, we have glutathione and GST fusion interaction. So these are only the types of interaction which are found in case of affinity chromatography. Are you able to get it? So this is only the affinity chromatography. Now look at the size of this ligand. So only these protein molecules, the blue one, they are allowed to bind, and the rest will be washed out. And then we have to, not only the binding is not the only thing, we have to do the elution also. We have to add the elution buffer also, because we want to collect the desired protein also. So if now this is the interaction between our desired protein along with the ligand. Now what we want? We want this protein back. So what we will do? We will add another ligand. So our protein will bind with this ligand and will be moved out from the column. This gel filtration we already done yesterday. So are you able to get my point basically? So this is only the gel filtration this is only our affinity chromatography. Are you able to get it? This is only the affinity chromatography. You have to revise, you have to remind this thing, that we have an immobilized ligand. So ligands are present on a stationary phase, or they are immobilized in nature. So that's all for affinity chromatography. We have one small video for affinity chromatography. Let's see it once, then we will move further to the flow cytometry. So this is affinity chromatography.
Here one quick biochemistry basics here. The chromatography technique in which separation occurs based on the specific interaction between the two molecules. This specific interaction can be between enzyme and a substrate, receptor and a ligand, protein and the nucleic acid, etc. Let's see different components of the affinity chromatography. The affinity chromatography is usually carried out in column, which is filled with the supporting material or the matrix. The matrix should have the following properties. It should be chemically inert, should exhibit good flow properties, should have sufficient functional groups for the covalent binding of the ligand. Different materials like agarose, polyacrylamide, polystyrene, cellulose, silica, etc. can be used as a matrix. The matrix is attached with a spacer M, which is usually made up of CH2 group. The Presence of spacer prevents the non-specific interaction of the ligand with the matrix itself. Let's see the working of affinity chromatography. When the mixture of different molecules is passed through the affinity column, only the molecules that have specific affinity for the ligand binds the ligand. The other molecules are washed away. Next, to disassociate the molecule of interest from the ligand, a buffer with different ionic strength or pH is passed. This finally releases the molecule of interest from the ligand and the molecule is obtained in the pure form. The molecule of interest can also be detached from the ligand by using a competitive inhibitor. The competitive inhibitor can further be removed by dialysis or by changing the pH or the ionic strength. Let's see different examples of ligand and the molecule of interest that can be purified with affinity chromatography. AMP can be used for the purification of NAD-dependent dehydrogenase and some kinases. ADP can be used for the purification of NADP-dependent dehydrogenases. Calmodulin for the purification of calmodulin-binding enzymes. Avidin can be used for the purification of biotin-containing enzymes. Metals such as nickel can be used for the purification of proteins that have a histidine tag. Lysine can be used for the purification of rRNA.
Okay, so are you able to get it? Are you able to understand affinity chromatography? Okay, so now we have to move with our another topic, which is flow cytometry. So have you done flow cytometry before? Have you listened about flow cytometry? Kindly let me know, please let me know. So we have to do flow cytometry today. So just waiting for your replies so that we will move further. Yes. Very good. So you are able to get the point of affinity chromatography, which is basically what is basically affinity chromatography, what is ligands, how they will attach with the specific receptors. You know about it. Okay, very good. Where is Anushri? Where is Juveria? Kindly reply, please. Yes, very good. Flowing cell counter, very good. So are you getting my point? Are you able to understand affinity chromatography? So now we have to move with flow cytometry. So have you done it before? Can you let me know? Yes, ma'am. Anushri saying yes, ma'am. Have you done it before, Anushri? Flow cytometry. Kindly let me know, please. Okay, so we have to start flow cytometry today, or kindly reply, please. Kindly. Where is Juveria? Where is? No, ma'am. Okay. That's why I just want to know whether you have done it or I have to start it from very basic. That's why I am asking again and again. Kindly reply me. Have you heard about the term flow cytometry? Have you heard about it or not? So that we will start it from a very basic, because it's a very important technique and it's a little bit complex technique also. It's a little bit complex because it's a very advanced technique. But we have some animation of flow cytometry also, you will be able to understand. Okay. So you have not done it. Okay. We have Tanmay here, Vaishnavi is here, Gayatri is also here. Yes, Gayatri, this flow cytometry is a little bit complex technique. It's not as easy as we have blotting or our this STS. It's not that much easy. So if you are not able to get it today, no issues, no worries. We will repeat it tomorrow also. We will discuss it thoroughly, and I will try my best to convey it to you. Okay, so no issues, no issues. So basically it's a technique, it is apparatus. Okay, let me start now. Let me allow to start it now because it's already okay. So we will start with flow cytometry. It's a little bit advanced technique. It's a little bit new technique for all of you. So if you are not able to get it, no issues, no worry. We will discuss it again and again, but you have to understand the basic concept only. So there are lots of applications of this technique, lots of, but our main focus is to know the basic of the technique. What is the function of a technique? What's the principle of a technique? How the technique works? This is our only motive. So I will start with the flow cytometry now. So if you are able to see this image, you can see we have a computer here in which we have some type of graphs, and we have a machine or a apparatus here. This is generally a flow cytometer. So what is flow cytometry? It is the measuring properties of a cell as they flow in a fluid suspension across an illuminated light path. So what happens? We have apparatus in which we allow the cell to move. So they are saying the cell, they flow in a suspension. So we have a chamber or a column in which the cells are moved, and we have a light path also. So they have to pass through this light path. I will show you. So this technique is a flow cytometry, is a recent technique. It exists in more than 5 decades now. So basically this technique was generally appeared in early 90s during AIDS epidemic, when the AIDS is on its prominent peak. At that time we have this flow cytometry which gets introduced. And there are also for another types of cells like CD4 T cells. These are the part of immunology. So we have different types of cells in our body. To analyze these cells, we have this technique. Now the main thing which we have to study. The method allows a quantitative and qualitative analysis of several properties of a cell. So if you understand what cell is, basically suppose this is a cell. Cell consists of nucleus. In cell we have mitochondria also. We have our own DNA also. We have cytoplasm also. Endoplasmic reticulum, Golgi body. We have the cell has its own proper structure. So we have to study the properties of a cell. So in flow cytometry, we are able to study the properties of a cell. First of all, remind this thing. From virtually any type of fresh unfixed tissue or a body fluid. So if we have a fresh live tissue, we are able to study the properties of that particular cell. The properties measured include a particles related size. So what we are able to do? We are able to study the size of a cell, that how much small or how much large the cell is. Like in our body, in our blood, in our blood we have different types of cells. We have neutrophils, we have basophils. You all know the composition of it. So flow cytometry is generally used to measure the size of a cell. Like we have 100 cells here of different sizes. How you will measure their size, or how you will count the number of cells? First technique is that you will see it under a microscope. But if you will see it under a microscope, it becomes very time consuming process. How will you count 1000 cells, or how will you count 10,000 cells? So for this purpose we have flow cytometry. So flow cytometry is used to know the size of a cell, number one. Then we have to know their granularity. What do you mean by granularity? Basically complexity. We have to know the complexity of a cell and relative fluorescence intensity. Three things we done in flow cytometry. Number one is we are able to know the size of a cell. Number two, the complexity or the granularity of the cell. Number three, relative fluorescence intensity of a cell. So basically we have taken the cells of a blood, bone marrow or lymph nodes. So in the blood we have basophils, neutrophils. So by these we are able to know the concentration or the number of cells. Flow cytometry is basically used to know the size of a cell, number of a cell, granularity of the cell and relative fluorescence intensity. I will show you somewhere. Like this is the place where you will put your sample and you will able to see the results. In this technique what happen, we have to do nothing. We have to put our sample here and we are able to see the results on the computer only, and the results are in the form of graphs. So this is the basic mechanism. First of all we will put our biological sample. The sample may be blood or your bone marrow, anything may be. Then you have to add some fluorescent marker. What is fluorescent marker? Which will shine when you will visualize it, it will shine. So then we have laser beam, we have apparatus by which it will pass. I will show you. So flow cytometry, as the name suggests, is a technique for cell counting. So you have to remember that for cell counting on how many number of cells are present. Number of cells, whether there are 5 cells are present, 100 cells are present, 50,000 cells are present or 1 lakh cells are present. So we have to know the cell counting. It's a technique for cell counting and measurement of different properties of a cell. What are the properties of a cell? Like its complexity, its granularity. So it is a laser based technology that measures and analyzes different physical and chemical properties of the cells flowing in a stream of fluid through a beam of light. So I will show you 1 minute. So this is the diagram. Basically this is the flow cytometry diagram. We have a sample which we load here. We have number of cells. If you are able to see the size of cell, the size of the cell is different, the shape of the cell is different. Like this one is big one and the upper one is small one. Are you able to get it? And the lower one is also small one. These are big ones. So what will happen? We will allow to pass from the nozzle, from the apparatus, and we have only a single cell during a detection. So by this we will analyze how many cells are present. Only a single cell will be allowed to pass, like 1, 2, 3. So this cell has passed. So the computer has collected this, that this is number 1. They recorded it, that this is number 1. Now this is number 2 cell. This is number 3. So by this we can do the cell counting. So we have a laser beam. Which will put on this sample, also on this sample, but now this move on. And we have some detectors present. Which will put the record here on the computer. So first I want to show you some basic things. So this is again, we have number of cells. You can see in the nozzle we have 1, 2, how many number of cells. But later on, only when one cell is allowed to pass on. So by this technique we are able to count the number of cells. So once the sample is injected into a stream of sheath fluid within the flow chamber, this is known as flow chamber, where we have number of cells. They are forced into the center of the stream, forming a single file. So only one single cell is allowed to pass down by the principle of hydrodynamic focusing. So only one cell or a particle can pass through this laser beam at a given moment. So at a single time, only a single cell is allowed to pass down by this hydrodynamic focusing. This is the number one point. So in flow cytometry, it's a technique for counting the number of cells, for knowing the internal structure, that is complexity, granularity of a cell, and also to know the fluorescency of the cell which is attached with the cell. So we have three important points in flow cytometry. So this is another point which we have to remember, that it works on a hydrodynamic focusing in which only a single cell is allowed to pass through a laser beam. Laser beam, when the cell comes in contact with a laser beam, we get the detectors which are present here. The detectors which are present here, they are able to collect all information, and it will come in our computer in the form of graphs. Like this is a graph, but I will tell you the basic things first. So in the flow system, one of the fundamental of flow cytometry is the ability to measure the properties of an individual particle or a cell. We will come to know about the cell count of a cell as well as the properties of a cell. So when a sample is injected into a flow cytometer, it is ordered into a stream of single particle. We have a central channel through which the sample is injected inside, and we have an outer sheet. It contains faster flowing fluid enclosing the central core. So basically this technique is used to know the cell count and to know the properties of the cell, to know the properties of an individual cell. So how the laser beam, how it affects the thing. So if you are, this is again the same thing, this one is the same, this one is. So only a single cell is allowed to pass. Only a single cell is allowed to pass, and we have a laser beam which will put it on this particle, and then we have a detector which are attached with a computer. Get it? So what will happen basically, when this laser beam put it on this cell particle or a particle which may be of different sizes, what will happen? It will scatter. It will scatter somewhere. If you can see here, not here, 1 minute, like here. So what will happen? This particle, this is a particle, this is a cell. If you are able to see, we have a nucleus in the center. So from here the laser beam comes. What happens? Some beams pass as such. Some light beams pass as such. But some gaps with the cell and they scatter in this direction. Are you able to get my point? अगर किसी चीज के साथ कोई बीम बजेगी तो क्या होगा? Some beams will pass as such. They will pass as such. But some beams will touch with this particle and scatter back. They will scatter back. So this has happened in this case. Light that is scattered in the forward direction, that the light which passed from it into the forward direction is known as forward scattering or forward scatter light. Are you getting my point? So we have two types of scattering. We have forward scatter and we have the side scatter. In side scatter what happens? Like this is forward scatter, the light will pass as such. In side scatter what happens? The beam touch the particle and they reflect somewhere else. They move somewhere else. So laser light that is scattered at 90 degrees to the axis of the laser path is detected in the side scatter channel. We have two types of channels. We have forward scatter and we have the side scatter. So by this scattering of light, we will able to understand the granularity or the complexity of the cell. This is the another point. So how many points we have discussed? we have discussed this thing that only single cell will allow to pass. Then we have, like similarly here, if you can see here, this is the beam of light which put on this cell. Some beams move as such, but some beams, what happen, like this is forward scatter, some beams pass as such, but some beams, when they collapse with the particle, they move on the different directions. Are you getting my point? So we have the forward scatter in which the beams are going forward, but when they collapse with the when they collapse the cell molecule or cell particle, some beams move on to the different directions. Now what happened with this? What happened with this scattering? What happened? So when the beam pass as such, you can see the beam pass as such in forward scattering, you will able to know the size of the cell. By forward scattering, you will be able to know the size of the cell, whether the size is small or whether the size is large. How? By the scattering. You can see how much scattering is here, and if the particle is large in size, how much scattering is here. Are you able to get my point? So you will able to see it in the graph. You will able to see it in a graph. So now this is a graph of forward scattering. What happens if the scattering is more? If the बच्चे जितना साइज बड़ा होगा, if the scattering is more, obviously scattering क्या होगी, ज़्यादा होगी. If the scattering is more, the size on the graph will be more also. So these are basically for the neutrophils. Lymphocytes, monocytes, neutrophils, basophils, these are all are the blood components. So by now the neutrophil is quite big cell, very big cell. What happen? They have the forward scattering is more here, and you will see it in the form of a graph. Then we have monocytes which are smaller than it. Suppose this is a monocyte. So the scattering is less, and we can see this pinkish. The rounded structure is only for your convenience only. So we can see here the size of the cell. If the molecule is very small, like lymphocyte, the scattering is very less, only very less. So you can see the representation here. So we have forward scattering and we have side scattering. Forward scattering is generally for to know the size of the cell, because we are able to know the intensity of light beam which pass through it. So we can get it in the form of a graph, and we will able to know the size of the cell, whether the cell is of small size or it is of a big size, of a large size. So with forward scattering we are able to get the size of the cell. Are you get my point? So we have forward scattering. With the help of forward scattering we are able to know the size of the cell. Let me show you in some another diagram. Like here also. So this is again a graph of forward scattering and side scattering. Right now I am just focusing on the forward scattering, which is the size of the cell. So here in this graph you can see these dots. These may be of different colors. So this large one, you are able to understand here the concentration of the graph is here, or the particle is here more. So they are generally granulocytes which are larger in size. Then we have another cells which are monocytes. So by FCS and FCS we are able to know, generally by forward scattering we are able to know the size of the cell, whether it is small in size or large in size. Number one point. And of course in blood we have RBCs, we have debris. They will just remain very less. Then we have granulocytes, monocytes, and we have lymphocytes. So we will get the graph like it. Number one point. So this is for the forward scattering. Now I have not discussed side scattering yet. I am just telling you about the forward scattering. In forward scattering, what happen, according to the size, we are able to get the, like, how much light pass through this. If it is small in size, you can know if the size is that much small, how much light will pass through it, only that one. So by this, like here the size of the particle and here the size of particle, so by this we are able to know the size of the cell, how much small is it or how much large is it. So this is the forward scattering. I have not discussed about side scattering, so this is only forward scattering. So forward and side scattering can be used to distinguish normal lymphocytes, granulocytes and monocytes. Basically, we are able to distinguish the size of the cell. The detection of lymphocytes and monocytes provides a reliable internal control to evaluate the size of the cells of the interest. So we will able to understand or to know the size of the cell of our own interest. So I want to show it again. So physical properties such as size. Size can be represented by forward scattering. So if you are, this is the cell, the light which passes as such, this is a laser beam which passes as such, this is known as forward scattering. So in this side we have forward scatterer on the graph or on the computer, and in this side we have a side scatterer. So what will happen when the beam will pass through this cell, we are able to know the size of the cell. So if you can see here in forward scatterer, this is the concentration, or by it we were able to know the size. So forward scattering is generally to know the size of the cell, and internal complexity is known by the side scattering. So the cell has some internal complexity also, like here also you can see the shape of neutrophils. The shape of neutrophil is little bit more complex as compared to monocytes, and the monocytes are little bit more complex as compared to lymphocytes. So complexity of the cell, the granularity or the complexity, that how much complex the cell is known by side scattering. We have two types of scattering, forward scattering and side scattering. Forward scattering is for to know the size of the cell, that how big or how small the size is, and the side scattering is generally to know the complexity or the granularity of the cell, that monocytes or lymphocytes, how complex they are in the inside. So internal complexity can be known by the side scattering. So in the graph we have forward scatter on this side and we have side scatter on this side. I have completed forward scatter. Now we will focus on side scatter. And then we have another point, forward scattering, side scattering. Then we have another point which is fluorescency, which we will discuss either today or tomorrow. So now we have to focus on side scattering. So in forward scattering, what happens? Forward collects light at 180 from the point at which laser beam intersects the cells, usually on a linear scale. What's the function of FSC or forward scattering? To know the size of the cell, and thus can distinguish between lymphocytes, which are small in size. You can see here, na? So lymphocytes are small in size. And then we have monocytes, then we have granulocytes. We have another types of cells. So we have neutrophils, we have monocytes, we have lymphocytes. So lymphocytes are small in size, monocytes are large, and neutrophils are very large. So with this we are able to distinguish the size of the cell, number one. This is by forward scattering. Now by side scattering, what happens? Side scattering collects right angle light at 90 and it's correlated with the cytoplasmic granularity, or you can say complexity of the cell, that how much complex the cell is internally or from the inside. So on this side we have forward scattering graph, or on the above side we have side scattering graph. So if you are able to see now, because I mark it so much, this one, one minute, let me discard it. So if you are able to see now, this one, now I am focusing on side scattering. I am not saying about the size of the cell. We are able to understand by forward scattering that the neutrophils are quite big in size, or these one are, you can see na, here we are moving. So here this is only a 50, so this is small in size, and here it comes under it 100. So this is small, then we have the bigger one, and then we have the larger one. So we have the lymphocytes first. We have, you know how to read on the graph. So we are moving on this side. So if we are moving on this side, the size will increase on this side. Suppose if something is here, then it is the bigger from all of these. So in this side or in this direction, the graph, the size increased. And on the upper side, the complexity or the side scattering increased. So if you can see here on this point, like here, we have small sized cells and they are little complex. They are small. Two things are together here. They are small and they are little in size. As we move forward, you can see here the size. We don't focus on size here. The complexity or the granularity is increased. So lymphocytes are less complex, monocytes are more complex, and neutrophils are complex in complex. Let me show you the diagram again. Let me show you. If you can see here now, by this direction, size of the cell is increasing, which we have discussed. Now we have to focus on side scattering, that is on this direction. On this direction, the complexity increased. So this structure or this cell is little complex because it comes under 50, and this large one comes under 250, so it is more complex. Suppose if in graph some cell is here, suppose. So what will happen? It becomes more increase in size and is more complex. This is the one situation. The another thing which will happen, suppose instead of increasing the size is small, but it is present here. So here what happens? Size is small, size is 50 only, but scattering is more. So complexity is high, but the size is small. Complexity is high, but the size is. We have forward scattering on x-axis, we have side scattering on the y-axis. These are the only things which we have to understand in case of flow cytometry. Now let me check whether you are getting it or not. Are you able to get it? Okay, Anushri, Juvelia, are you getting it, bache? You are understanding the basic concept of flow cytometry. All of you kindly reply. It's very important for me to know. Like Anushri, Rahul, Sakshi, Saurabh, Tanmay, Vaishnavi, Gayatri, please let me know. Are you able to understand flow forward scattering and side scattering? Are you able to get it? Juvelia, Rituja, Hital, Saurabh, kindly let me know, please. Yes, Hital is there. Rahul is saying yes, ma'am. Siddhi is here. So please, all of you kindly let me know. Are you able to get it? It's a very recent technique, but I just tell you about the basic principle or the basic concept only. We have forward scattering, we have side scattering, and this technique is used for cell counting, the number of cells, how many number of cells are there, the size of the cell, and the internal complexity of the cell. So kindly reply Vaishnavi, Priya. Then we have Rutuja, Gayatri. Reply, please reply. Yes, Aditi is here also. Okay, so this is the one aspect I have not touched yet. I have touched all the things, but one thing is left yet. But let me see whether you are getting it or not. Where is another student? Chloris is here. Okay, very good. Shubham is here. Very good, very good. So are you able to get it? Now I will show you small animation of this whole technique. Let's see the animation then.
We will introduce you to the flow cytometry experimental technique. Flow cytometry is used to count and analyze the size, shape, and properties of individual cells within a heterogeneous population of cells. Flow cytometry data is extremely quantitative and can be analyzed in depth by specific flow cytometry software programs. Once a suspension of individual cells has been prepared for flow cytometry analysis from cells, tissues, or organisms, the prepared sample is placed on the flow cytometer. How does a flow cytometer analyze the size, shape, and properties of individual cells? A flow cytometer contains several key components, including the sample, fluidics that move the sample into the flow cytometer, lasers, optics which gather the light, detectors to sense the light, and a computer system to output the data into a form that can be analyzed by the researcher. Once a sample has been placed in the flow cytometer, the machine will suck up the sample, mix the sample into a stream of saline solution in the cytometer, and lead the cell suspension through a narrowing channel, causing the cells to form a single file line before the cells pass through the laser at the interrogation point. Each cell within the sample then moves across the laser beam, allowing each cell within the sample to be individually analyzed. As each cell passes through the laser beam will scatter in multiple directions. The flow cytometer detects light scattered in a forward manner, called forward scatter, and light scattered in a sideways manner, called side scatter. The amount of forward scattered light for each cell is detected by a detector on the far side of the cell from the laser. Forward scatter is proportional to the size of the cell. The detector converts the scattered light into a voltage pulse, which is directly proportional to the amount of forward scattered light. The computer converts these data into a histogram plot with the amount of forward scattered light on the x-axis and the number of cells on the y-axis. The amount of side scattered light is detected by a detector located perpendicular to the path of the laser beam. Side scatter is proportional to the shape and internal complexity of a cell. Just like with forward scattered light, the flow cytometer converts the detected side scattered light into a voltage pulse, which is directly proportional to the amount of side scattered light. By analyzing the forward and side scatter data together, the researcher can understand a cell's size, shape, and complexity. In addition, the analysis of the forward and side scatter data can allow the researcher to divide the heterogeneous population of cells into individual populations with varying size, shape, and complexity. This capability of analyzing multiple populations within a sample is the immense strength of the flow cytometry experimental technique. In addition to analyzing a cell's size, shape, and complexity, a flow cytometer can also detect emitted light from excited fluorescent molecules, such as fluorescently labeled antibodies or fluorescent dyes or stains. For the rest of this animation, we will focus on the detection of fluorescent signal by the flow cytometer. For more information about fluorescence, please see the fluorescence animation. The fluorescent molecules, or fluorophores, are excited by the correct wavelength of laser light when the cell passes through the laser beam. After excitation, the light emitted by the fluorophore is directed along a path with emission filters that allow for the detection of multiple different fluorophores emitting light in a cell. A flow cytometer can detect the presence of a fluorophore and also quantify the relative amount of a fluorophore within a cell. A flow cytometer can also be used to detect light emitted from multiple fluorophores in the same sample. While it is common for flow cytometers to detect 2, 3, or 4 colors at the same time, some flow cytometers can even detect up to 18 different colors at the same time. The data can then be presented to the researcher in the form of a histogram or a dot plot. Such data furthers the powers of flow cytometry analysis by enabling the researcher to analyze individual populations of cells within a heterogeneous mix of cells in the sample.
So I hope you will able to understand flow cytometry, the forward scatter and the side scatter, and another thing which is left is the fluorophore. Now we will discuss it now. So are you able to get it from the video also? Only one small topic is left, which is the fluorophore. Let me discuss it, and by this we will complete the flow cytometry. So kindly reply, are you getting it? Okay, I hope you are getting it. I hope. So Bhagyashree is also here. Yes, very good, very good. So only one part is left. Let me conclude it then. And now I am going to the presentation. So the main thing is the size of the cell, whether the size is small or large. It can be known, or we can detect it with the help of forward scattering, and then we have the side scattering, by which we can know the complexity of the cell, that how complex the internal structures are. So we have to run the graph. In this side we have forward scattering, and on this side we have a side scattering. Then these things we have done hydrodynamic focusing, we have done it. Then we have done side scattering. This is the laser beam which passed, and we have to detect it on a detector. Then we have done, like here you are able to see, like if forward scattering is increasing, that the size of the cell is increasing, the complexity is also increasing. The complexity of the cell is also increasing. So both SS and SC, it's not necessary that they will move as such. No, sometimes what happens, the size of the cell is very small, like it's only 15, but they are more complex. So it's not necessary that with the forward scattering and side scattering are proportional. No, sometimes what happens, the size of the cell is small, but they are little bit more complex. So this is your flow cytometry, and they are generally used in immunophenotyping, cell sorting, to separate the different types of cells, to know the DNA content in a cell, to know apoptosis, whether the cell is going under any type of disturbance inside. So for apoptosis, for cell cycle analysis, and for cell proliferation assays. So these are the applications of flow cytometry. Now we have another one thing which is left, which is the fluorescence. That is why we have known it as fluorescence activated cell sorting. From the name you will able to understand that the cells are sorted. Sorted means separated. The cells are separated or sorted on the basis of fluorescence. Now what's the fluorescence? The color which shines, fluorescence color. So cells are sorted. It's a fluorescence activated cell sorting. The cells are sorted according to the fluorescence they receive. So we have discussed it. Like they are just giving us example. Consider a group of lymphocytes from a mouse that have been stained with a green fluorescent antibody specific for CD4. So we have a fluorophore, which is fluorescent isothiocyanate, and red fluorescent antibody specific for CD8. So we have two fluorescents, we have green and we have red fluorescents. The green one is for CD4 cells, and the red one is for CD8 cells. We have two types of fluorescence. Both the labeled cells generate side scattering and forward scattering as they pass through the laser beam, creating voltage pulses that recorded by the computer. So both the cells, the CD4 cell and the CD8 cell, when they pass from the laser beam, they will show side scattering as well as forward scattering. We know about it. However, Each labeled cell will also emit light of specific wavelength. As we know, CD4 has green fluorescence, or you can say green fluorescent material. So each labeled cell will also emit light of specific wavelength as a result of fluorescent labeled. We have fluorescence attached with it. So CD4 cells will emit green fluorescent light of wavelength 525 to 530 nanometer, while CD8 cells, the CD8 cells will emit orange or reddish light of wavelength 560 nanometer. So as in the animation we also seen that the flow cytometry or the apparatus is able to detect 12 different or 12 different types of colors or fluorescent colors. So these fluorescent signals pass through the photo multiplier tubes and generate voltage pulses. The software integrates all the information for a particular cell, allowing the characterization of an individual cell. So by this technique we will be allowed to know the characterization of an individual cell or the characters of an individual cell, either it's a CD4 cell or a CD8 cell. So that is fluorescence activated cell sorting, in which we sort the cell by using some fluorescent chemical or also known as fluorophore. I will tell you here also. So like here we have different cells on different wavelength, like on 585 FL2 we have FL1, then we have FL3. These are on specific different wavelength. These are known as fluorochromes. Fluorochromes are those substances, like you can see this is a cell and just we add the fluorochrome here, the color that can be excited by a certain light source such as laser and emit a fluorescent signal at a single wavelength. So we will be able to, rather the cell is able to emit a fluorescent signal at a single wavelength of a specific wavelength. Like everyone has a, like this fluorochrome has a wavelength at 488. At this wavelength it will emit or it will start emitting the color. So fluorescent dyes can directly bind to certain cellular contents such as DNA or RNA and allow us to perform quantitative analysis on individual cells. However, in most cases, fluorochromes are conjugated with monoclonal antibodies which specifically target cellular markers. So they can attach with antibodies also, or they can attach with DNA or RNA also. But basically fluorochromes are forward purpose for quantitative analysis of the cells. So these are the fluorochromes. We have fluorescein, we have phycoerythrin, we have red, PE red. Then these fluorochromes, they work on a specific wavelength. That in this range they will emit the color, or in this wavelength from 633 to 767 they will emit a color, or you can say we will able to characterize the cell particularly. So interestingly, although some of them can be excited by same light source, the different fluorochromes may emit fluorescent signals with different wavelengths and different colors. So wavelengths are also different and the emission of color is also different, like green, red, orange, yellow. Thus multiple fluorochromes can simultaneously excited by a light source and detected by the emission of fluorescent signals. So with the help of this fluorescence or with the help of this fluorochromes, we are able to detect the fluorescent signals which the cell gives us with different wavelengths respectively. So with the help of fluorochromes we are able to detect the fluorescent signals which are at specific wavelength, and the color is also specific. So if you can see here, we are adding different, like in for killer T cell spores the fluorochrome gives purple color, and for helper T cell the fluorochrome gives us the red color. So what will happen when it will pass through it? What will happen? We will see this graph. We have forward scattering and side scattering, and also we have the colors which are here, like this red one. this is here. So that means the size of the cell is small and the complexity is also less because it is on the lower one. So this is the red one is which one? The helper cell. So these are the helper cells whose size is small and whose complexity is also less. Now suppose these are killer T cells. So we are able to see the purple color here. So what happens? The size of the cell is large as well as the complexity is also more. So what are these cells? The purple cells are what one? The killer cells. So we will able to know the type of cells, like either they are according to the color they carry, we are able to know whether they are helper cells, they are killer T cells, they are CD4 cell or they are CD8 cells. So if you can see here in this case CD4 and CD3, like this red one, suppose these are helper T cells, what will happen here? The complexity is also high and they are little bit complex in structure as compared to this green one or compared to this purple one. So by this comparison we are able to detect the cells particularly. So this is known as fluorescence activated cell sorter. With the help of these cells we are able to understand with the help of these fluorochromes we are able to understand the characterization of the cell, or basically what cell it is basically, because each cell will carry a specific color at specific wavelength. So these are the fluorochromes which we have used, which we will use basically, and the fluorochromes they attach to the cell surface, or they can bind to the DNA or RNA, or they can even bind with the antigen or the antibodies which are present in the cell. So this is all for flow cytometry. I hope you will get it. So I hope you are able to get it what I want to convey. Amita is saying, so how that can appear in graph, like if it is small then present at the bottom and more complex then it has to be present on the top. Actually I am saying that side scattering and forward scattering are individual only. Side scattering has its own function, forward scattering has its own function, side scattering has its own function. So we have not to judge it. They are actually both are a different graphs, but just we have to combine it singly. If you can see in the animation also, they are the different graphs, but we combine it in a single graph. So the results which are given by side graph or the side scattering is totally different from the results which are given by the forward scattering. Suppose it's not necessary if the size of the cell is big then it becomes more complex. No, it's not needed. Sometimes the size of the cell is small, that is they are forward scattering, but they are more complex in nature. So you have not to confuse side scattering and forward scattering together. No, they are different. They have the different graphs and they have the different properties on which they are working. If the size of the cell is small, it doesn't mean that it should not be complex. Maybe it's complex, but it is small in nature. So what is wrong in it? Nothing is wrong in it. And sometimes what happens, the size of the cell is quite big, but still its complexity is very less. So you have not to confuse side scattering and forward scattering together. No, they are totally different from each other. They have different functions. Side scattering is for complexity of the cell and forward scattering is for the size of the cell. So both are different. You have not to mix it together. So Amita, are you able to get it? बच्चे समझ आई आपको? Amita, are you able to get it? So you have not to confuse side scattering and forward scattering. Side scattering, the functioning of the side scattering is different and the functioning of the forward scattering is the different. You have not to mix the properties of both two. Okay, are you able to get it? If the size of the cell is small, so in the forward scattering it will come first, but it doesn't mean that it is not complex. Maybe it's more complex. Yes, if it is more complex, then it will come in the side scattering. It will come in on the upper side. But the presentation which I am saying, I show you the side and the forward scattering are on the same graph. So that's okay, you are saying, right? If it is small, then how it is present in the bottom, or if it is complex, then it should be on the top. So you will make the graph by your own self, but you have to understand the concept only, that if it is small, maybe it's a complex, graph of the side scattering and the forward scattering are totally different. You have not to merge it, or you have not to mix it. So are you able to get it now? Please let me know. Come on, come on. Where are Rituja? Where is Anushri? Amita? Aditi? Okay, it's very, you have to remember 2-3 points in your mind. You have to remember the basic concept, that for what thing flow cytometry is basically for, to know the size of the cell, to know the complexity of the cell, and then we have fluorochromes are here to know, by using the fluorochromes we are more able to understand the type of cell which is present, either it is a T cell present, or a helper cell present, or a killer cell present. So with the help of fluorochromes or fluorescent, we are able to recognize the cell more authentically. Okay, so now we have one more little presentation or animation. After it, the flow cytometry will be completed. So let me show it. It's a very small video for your understanding basically.
Diploidy analysis to cell counting and GFP expression analysis. Cytometers are used in a range of applications, from immunophenotyping to ploidy analysis to cell counting and GFP expression analysis. The flow cytometer performs this analysis by passing thousands of cells per second through a laser beam and capturing the light that emerges from each cell as it passes through. The data gathered can be analyzed statistically by flow cytometry software to report cellular characteristics such as size, complexity, phenotype, and health. In this tutorial, we will look at how a flow cytometer works, how scattered light and fluorescence are detected by a flow cytometer, and how the resulting data can be analyzed. This view shows the primary systems of the flow cytometer schematically. These are the fluidic system, which presents samples to the interrogation point and takes away the waste; the lasers, which are the light source for scatter and fluorescence; the optics, which gather and direct the light; the detectors, which receive the light; and the electronics and the peripheral computer system, which convert the signals from the detectors into digital data and perform the necessary analyses. The interrogation point is the heart of the system. This is where the laser and the sample intersect, and the optics collect the resulting scatter and fluorescence. First, let's talk about how the sample is delivered to the laser. Here we see how the sample is transported through the interrogation point. For accurate data collection, it is important that particles or cells are passed through the laser beam one at a time. Most flow cytometers accomplish this by injecting the sample stream containing the cells into a flowing stream of sheath fluid or saline solution. As you can see, the sample stream becomes compressed to roughly 1 cell in diameter. This is called hydrodynamic focusing. In fact, flow cytometers can accommodate cells that span roughly 3 orders of magnitude in size. In most cases, cytometers will be detecting cells between 1 and 15 microns in diameter, although through the use of specialized systems, it is possible to detect particles outside this range. Now let's see how laser light is used to detect individual cells in the stream. As a cell passes through the laser, it will refract or scatter light at all angles. Forward scatter, or low-angle light scatter, is the amount of light that's scattered in the forward direction as laser light strikes the cell. The magnitude of forward scatter is roughly proportional to the size of the cell, and this data can be used to quantify that parameter. But how can we record this scattered light? Light is quantified by a detector that converts intensity into voltage. In most cytometers, a blocking bar, called an obscuration bar, is placed in front of the forward scatter detector. The obscuration bar prevents any of the intense laser light from reaching the detector. As a cell crosses the laser, light is scattered around the obscuration bar and is collected by the detector. The scattered light received by the detector is translated into a voltage pulse. Because small cells produce a small amount of forward scatter and large cells produce a large amount of forward scatter, the magnitude of the voltage pulse recorded for each cell is proportional to the cell size. If we plot a histogram of these data, smaller cells appear toward the left and larger cells appear toward the right. A histogram of forward scatter data is a graphical representation of the size distribution within the population, but such a graph only presents one-dimensional data. Next, let's look at side scatter. As we have already seen, a cell traveling through the laser beam will scatter light at all angles. Light scattering at larger angles, for example to the side, is caused by granularity and structural complexity inside the cell. This side-scattered light is focused through a lens system and is collected by a separate detector, usually located 90 degrees from the laser's path. The signals collected by the side-scatter detector can be plotted on one-dimensional histograms, like we saw for forward scatter. The one-dimensional histograms we have seen so far do not necessarily show the complexity of the cell populations. For example, what appears to be a single population in the forward scatter histogram is, in reality, multiple populations that can only be discerned by looking at the data in a second dimension. This is done through the use of two-dimensional dot or scatter plots. You can see that the peaks from the forward and side scatter histograms correlate with the colored dots in the scatter plot. Now we can view the results obtained when we create a scatter plot using forward and side scatter data from a typical peripheral blood cell run. The populations that emerge include lymphocytes, which are small cells possessing low internal complexity, monocytes, which are medium-sized cells with slightly more internal complexity, and neutrophils and other granulocytes, which are large cells that have a lot of internal complexity. This multi-parametric analysis is the real power of flow cytometry. Now let's take a look at another parameter that can tell us more about cell structure and function: fluorescence. As a brief review, fluorescence is a term used to describe the excitation of a fluorophore to a higher energy level, followed by the return of that fluorophore to its ground state with the emission of light. The energy in the emitted light is dependent on the energy level to which the fluorophore is excited, and that light has a specific wavelength and consequently a specific color. One of the most common ways to study cellular characteristics using flow cytometry involves the use of fluorescent molecules, such as fluorophore-labeled antibodies. In these experiments, the labeled antibody is added to the cell sample. The antibody then binds to a specific molecule on the cell surface or inside the cell. Finally, when laser light of the right wavelength strikes the fluorophore, a fluorescent signal is emitted and detected by the flow cytometer. How is this fluorescence information collected? The fluorescent light, coming from labeled cells as they pass through the laser, travels along the same path as the side scatter signal. As the light travels along this path, it is directed through a series of filters and mirrors so that particular wavelength ranges are delivered to the appropriate detectors. Fluorescence data is collected in generally the same way as forward and side scatter data. In a population of labeled cells, some will be brighter than others. As each cell crosses the path of the laser, a fluorescence signal is generated. The fluorescent light is then directed to the appropriate detector, where it is translated into a voltage pulse, proportional to the amount of fluorescence emitted. All of the voltage pulses are recorded and can be presented graphically. What if we want to do a two-color experiment? We need to look at the spectra of the two fluorophores to see if they are compatible. Alexafluor 488 and phycoerythrin, or RPE, are commonly used together. For these two fluorophores, 488 nanometer light is an efficient excitation source. When excited with 488 nanometer light, you can see that the emission peaks for these two dyes are far enough apart so that discrete emission data can be collected. Compatible dyes, such as these, allow scientists to easily detect two colors from a single laser. If we analyze data from the two-color experiment using a scatter plot, four distinct populations emerge. Looking at the dot plot in terms of quadrants, Cells with only bright orange fluorescence appear in the upper left quadrant. Cells with only green fluorescence appear in the lower right quadrant. Cells with both bright green and bright orange fluorescence appear in the upper right quadrant. And finally, cells with both low green and low orange fluorescence appear in the lower left quadrant. Multiple fluorescence parameters are necessary to dissect complex biological systems. How does the flow cytometer collect discrete fluorescence data for the Alexa Fluor 488 and RPE fluorophores? Filters are available that can capture the peak fluorescence from each of these molecules. A 530 nanometer bandpass filter will collect most of the Alexa Fluor 488 peak, and a 585 nanometer bandpass filter will collect the bulk of the RPE peak. Using these filters, the proportional amounts of Alexa Fluor 488 and RPE fluorescence can be recorded for each cell. You can see that portions of each emission peak overlap one another. This is called spectral overlap, and we will talk about how to deal with this in the next tutorial. Let's take a moment to talk about filter nomenclature. Filters are normally defined by one of two parameters, either the center point for a bandpass filter or the cutoff point for a long or short pass filter. This is a typical bandpass filter specific for RPE. It has a center point of 585 nanometers.
So I hope you all will understand the flow cytometry. There are lots of other functioning or applications of flow cytometry, but we have to stick to the basic principle. The functioning of forward scatter, side scatter, and the fluorochrome. So that is all for the flow cytometry. If you have any type of query, kindly let me know. Are you able to get what we have discussed today, what we have read today? Are you able to understand it? Kindly reply if you are able to understand the things. Because flow cytometry is a very recent technology, it's a very recent technique. Nowadays it becomes prominent. So you have to understand, you have to remain with the basic principle, with the basic concept of the flow cytometry. So Anushri is here, Pooja is here, Juvelia, Debita, Siddhi, Shubh, Bhagyashri is here. Yes, okay, very good to all of you. So today's lecture is little bit, lengthy, but we have to cover the flow cytometry. It's very important. It's a very recent technology. Then, if you understand the basic concept, and there is only a graph representation in the computer or on the computer, so you will modify your results. You will get the results in the graphical form. So you can add more and more things in flow cytometry. But these are the basic things. These are basic things which we have to know. So Devita is saying, what's the timing for tomorrow? We will meet at 2 p.m. after lunch. So I will share the link with all of you. So be prepared for 2 p.m. From 2 p.m. to 4:30 or 5 p.m. How long it will take, I don't know. But I will try to finish 2 topics tomorrow. Because tomorrow is Sunday, we can do it. So yes, from 2 to 5 we are here together with all of you. So I just want to thank all of you that you are with me all along for 2 hours. So it's a very big thing, it's a very good thing that you are here properly with full concentration, with full energy. So I want to thank all of you. If you have any type of query, kindly let me know, and kindly also like the video, because it takes lots of efforts to collect the data and to collect the things. I just request all of you to kindly like it, or you don't like it, kindly let me know if there is some type of more things which we could add. If you have some type of suggestions, then you are most welcome. If you have some type of suggestions also, but tell me if you have some type of queries or something, please let me know. So that's all for today's session. I want to thank you all for superb energy and full concentration. I want to thank you all. Tomorrow we will meet. Maybe we will take the break in between for 10 minutes, for 5 minutes, for a little bit of refreshment. So tomorrow we will meet. Definitely we will meet. So that's all from my side. So is it okay? Ankita is saying thank you ma'am with smiley also. Thank you dear, thank you. We have Devita, we have Gayatri, Siddhi is here, Nupur is here. Nupur, okay, thank you to all, thank you to you. Vandana is here, Devashish is here. Very good. Nupur is saying thank you ma'am, thanks to you dear, thank you. So we have, if some queries are there, if you are not able to understand any point, sometimes it happen when we are doing the things so concentratedly, sometimes even the small thing is we are not able to get it. So you can message me at any time if you have any type of query, because sometimes it happens. So you shouldn't have any type of query in your mind, you have to ask me. So you can even personally message me, or even in a group you can message me. If you have any type of doubt, a little bit of doubt, then we will share, okay? So no need of worries, no need of any type of tensions. We will meet tomorrow. If anyone will not be there, then we will discuss the topic again also, or you can watch the video also. So you have not to take any type of tension or a worry. We are always there for you. So that's all from my side. So thank you all. We will meet tomorrow at 2 p.m. So should I go now? So okay, then we will meet tomorrow. Bye bye from my side. Bye Ankita, bye. We will meet tomorrow. So all the best, take care of yourself. If you have any type of query, you can ask me, you can message me in group, or you can message me personally also. So okay, bye. Prachi saying bye, Rahul bye, Vishaka bye, and she saying thank you. Bye. Good night to all of you. Good night. Take care of yourself. Please. So that's from my side. Nupur, bye Nupur. Vandana, bye ma'am. Ankita, good night. Devashish, bye. So take care of yourself and kindly recap the main points of flow cytometry. We will meet tomorrow sharp at 2 p.m. So thank you all.